making the biggest discoveries in science. They're all based on the work of others. You don't work in a vacuum, and all the science always builds on itself.
At UCSF, we work across boundaries with the collaboration of people in every department, and being able to do that allowed me to make the discoveries that I've made.
I think that progress in science often happens at the intersection between research areas, between fields. My name is Lilian Chong, and I was a graduate of the biophysics program at UCSF in 2002. And currently, I am a professor at the University of Pittsburgh. I have a very unusual lab in the Department of Chemistry because we don't have chemicals in our lab. It's all computers. What we do is we run physics-based computer simulations to look at how things work at the atom level. There's no way that we could build a microscope in the lab that could give you those detailed views, so we have to use computers to do it. The role that my lab has played in advancing this field is we have some advanced simulation methods that we've developed called WESPA that allows you to greatly extend the timescales. And we can get to femtosecond time steps, which is a million billionth of a second, or timescales as slow as the seconds and possibly beyond. Running the simulation doesn't take that much time, but then we have months that we're spending analyzing terabytes of data to explain how things really work at the atom level.
So I'm Leslie Benet. I graduated from UCSF with a PhD in pharmaceutical chemistry in 1965. And I've been a part of UCSF as a faculty member for 57 years, and the impact of my research is primarily related to drug dosing, how to correctly dose drugs in patients. I was the first person to actually show that intestinal metabolism was something that was a barrier to drug absorption. Because to get an effect from a drug, you have to have the right exposure to the various organs in the body, so that gives you the right response that's efficacious and not toxic. Understanding what are the parameters that you need to know and how you adjust them to make the right dosing decision. And it impacts all drugs, and it impacts all clinicians in terms of their drug dosing strategies. I advised the FDA, I advised companies, I ran the clinical studies to show that generic drugs give the same effects as the brand name drugs. And I am frequently a consultant to the industry in this area.
My name is Sue Acton. I graduated in 1991 with a PhD in pharmaceutical chemistry from UCSF. And I gained foundational knowledge that allowed me then to move to the East Coast in Boston, where I worked at MIT as a postdoc. There I discovered the HDL cholesterol receptor with my colleagues at MIT, and high-density lipoprotein, or HDL, is known for taking excess cholesterol out of the body. The discovery of the HDL cholesterol receptor was very exciting because this discovery has allowed us to understand how cholesterol moves in the body and why it's important to keep cholesterol down. Then I went into the pharmaceutical industry, into a biotech company called Millennium Pharmaceuticals, and we discovered ACE2. This is a protein that's involved in a system that controls blood pressure. And later, that protein, ACE2, was determined to be the receptor for the SARS-CoV-2 virus. We don't really know why the virus chose ACE2 to attach to, but we know that that protein helped the virus get into cells and thereby cause damage. And this happens in science, where a discovery from a long time ago can now bring new importance because the knowledge builds upon itself, and you never know when your discovery is going to be the one that's actually the target itself or leads to the next target.
During the pandemic, my lab got involved in a great international collaboration where we contributed our WESPA software and methods to enable these landmark simulations of how the spike protein on the coronavirus opens up. And on the front page of the New York Times in December 2021 is a movie of the simulation. At least 4 different labs were involved in this, and we generated a large set of pathways for the spike opening process. Right now is a really exciting time to be in my field of molecular simulations because I really think that we're beginning a golden age for this work, relevant to drug discovery and protein design and all these other fundamentally interesting biological processes out there.
One of the advantages of being a professor is not only the enjoyment of your discoveries, but being able to translate that and to teach it to your mentees. And it is the thrill of my life to be able to have the impacts both in research and teaching and to have the influence on drug development and regulation throughout the world.